नमस्ते बच्चों, चलो आज एनाटॉमी ऑफ फ्लावरिंग प्लांट्स चैप्टर को एग्जाम स्टाइल क्वेश्चन से मास्टर करते हैं। बिल्कुल रैपिड रिवीजन। रेडी हो? लेट्स डाइव राइट इन।
Alright, so imagine you've got a slide under the microscope. How do you tell if it's a dicot or a monocot? This is a classic question, right? To figure this out, we've got to go back to the basics first. Let's build it from the ground up. So, Section 1, the basics. Look, to understand any plant properly, you have to know its building blocks, right? I'm talking about the tissues. Everything, and I mean everything about identifying a plant starts right here. Okay, bacho, this is super important. Always remember this. A plant basically has just these three tissue systems. That's it. Think of the epidermal system as its outer skin, for protection. The ground tissue, well, that's all the main stuff inside, the bulk of it. And the vascular system, that's the plumbing, the transport pipelines, xylem and phloem. Here we go, a very, very common NEET question. What's the shape of the guard cells in the stomata for dicots and monocots, specifically grasses? This is a dead giveaway, a direct way to tell them apart. The answer's on the next slide. Pay close attention. See, it's that simple. In dicots, they are bean-shaped, like a kidney bean or rajma. But in monocots, especially grasses, they're dumbbell-shaped. Just remembering this tiny little detail could literally get you 4 marks. Easy peasy. Now, this is another huge clue, the whole idea of open versus closed vascular bundles. Open just means there's room for more growth because it has cambium. Closed, on the other hand, means no cambium, so no more secondary growth. It's fixed. And this, right here, is a fundamental difference between dicot and monocot stems. Remember that. All right, Section 2, the root investigation. Okay, let's dig in, literally. We're going to look at the roots now. So, what's the deal with dicot versus monocot roots? Let's find out. Okay, so how do we tell dicot and monocot roots apart? Just by looking at their vascular bundles and pith. Butcho, listen up. This is super important for those diagram-based questions. They'll just give you a cross-section in the exam and ask, identify this. So let's compare them side by side. All right, take a good look at this table. In a dicot root, you'll see fewer xylem bundles, usually just 2 to 4. We call this condition diarch to tetrarch. And the pith? It's tiny. Sometimes you can barely see it. But in a monocot root, there are many bundles, more than 6. That's called the polyarch condition. And the pith is large and very well developed. This is a key difference. Note it down. So, are we clear on roots? Good. Let's move up the plant now to the stem. And you know what? The differences here are even clearer, even easier to remember. So, what's the single biggest, most obvious feature to identify a dicot versus a monocot stem? I mean, if I gave you a random slide, what's that one thing you would look for to know the answer? Instantly. What do you think? You got it. It's the arrangement of the vascular bundles. Just by looking at their pattern, you can tell what you're looking at. Let's see exactly how. And here it is, the biggest difference of them all. In a dicot stem, the vascular bundles are neatly arranged in a ring, and they're open. But in a monocot stem, they're just scattered all over the place, and they're closed. But just seriously, just remember these 2 words, ring for dicot, scattered for monocot, and your 4 marks are pretty much guaranteed. Okay, let's move on to our final part, the leaf. We're going to investigate the leaf now. And just like with the root and stem, there are some very clear-cut differences here that are often asked in NEET. So when it comes to a dicot or dorsiventral leaf versus a monocot or isobilateral leaf, what's the story with their stomata and mesophyll? Direct questions come from this part, so let's break it down. Here's the crucial point. In a dicot leaf, you'll find most of the stomata on the lower side. Plus, its mesophyll, that's the ground tissue, is clearly differentiated into 2 layers, palisade and spongy parenchyma. Now, in a monocot leaf, the stomata are on both sides, almost in equal numbers. And the mesophyll? It's not differentiated at all. See? Super simple. And here's a special point, important for NEET. Have you heard of bulliform cells? Butcho, these special cells are found in grasses, which are monocots. Their job is amazing. When there's a water shortage, these cells help the leaf curl inwards to minimize water loss. It's a really cool and important adaptation. Okay, we've covered a lot of ground. So now let's look at some super tricks for revision, just to make sure all this information sticks in your brain for good. For the stem, just memorize this simple line. Dicot ring में है, monocot बिखरा हुआ है. Dicot vascular bundles are in a ring, and monocots are scattered. That's it. That's the trick. Think of this slide as your final cheat sheet, your short notes. Go ahead, take a screenshot. For the root, you count the xylem bundles. For the stem, you look at the pattern. And for the leaf, you check the inside filling, the mesophyll. That's all you need to do to identify them. All right, final challenge time. A plant section shows vascular bundles that are scattered, conjoint, and closed. Plus, each one is surrounded by a sclerenchymatous bundle sheath. Come on, Butcho, put your thinking caps on. What is it? Focus on those keywords: scattered, closed, bundle sheath. What does that point to?
And the answer is a monocot stem. If you got that right, well done. You see, the moment you see the words scattered and closed, the answer becomes crystal clear. And that's exactly how you crack questions in NEET. You catch the keywords and move on.
So there you have it, all the key differences in a nutshell. I really hope this quick revision was helpful for you. Now, as you practice, I want you to think about this next question. We know dicots can become woody over time. So what are the secondary changes that make that happen? Think about how secondary growth adds a whole new layer to this story. Keep practicing, and all the best for your exams.